Hi, living environment friends. So today I'm going to do a little bit more of like a lecture, and I'm going to teach you about these two things called transcription and translation. And this is for Wednesday, March 25th. This is the assignment, one part of the assignment for today. So when I film these kinds of videos with my AP class, I always like to start with something funny. So I'll start with like a meme or a song. But today I wanted to start with these Jordans I just got because I'm not going to be at school and you're not going to be able to see them. So I wanted to show you in person. Look how nice they are. I'm really sad you guys can't see them in real life because they're beautiful, but I just wanted to share with you my new shoes. So I'm going to go over what you should know at this point. I know there's a lot of information and some of you might not feel like you're learning that much, so I'll always try and refresh what you should know each day at the start of the video. So you should know that DNA is found in the nucleus of all your cells. Every single cell in your body, your eye cells, your cheek cells, your heart cells, they have a nucleus, and that nucleus has DNA inside of it. All your DNA is in your nucleus, okay? Your DNA is wound up. It's that really interesting, like, double helix, that ladder shape. It's really, really long, so it gets wound up into these chromosomes. Those little X-shaped things are called chromosomes. So you have 46 chromosomes in your nucleus. Unless we're talking about a sperm cell or an egg cell, then how many chromosomes does it have? 23, good. So hopefully you know that by now. The newest thing we've learned is that DNA molecules are made of 4 bases. They're also made of a sugar and a phosphate, but we're going to focus on the nitrogen bases, which are adenine, sorry, my cats are being so annoying, thymine, guanine, and cytosine. So those are the 4 bases. You should know that adenine pairs with thymine, A goes with T, and guanine pairs with cytosine. So I have this little song I wrote myself to help you guys remember it, and if you were in class, I would make you sing it, but you're not, so you're just going to follow along with my song. So here's how it goes. C goes to G, T goes to A. This right here is my DNA, base pairs. So that's my song. You can sing it if you want. It does help you to remember it, I promise. Alright, so we're going to do a little DNA practice together. I mean, yes, it's just me talking, but hopefully you're trying to answer as I go. So you're going to complete the DNA double strand below. So this is one strand of it right here. And then we're going to fill in the missing parts of each. Okay, so this is C. C is going to pair with which letter? Remember, C goes to G. And if C goes to G, that means T goes to A. This right here is my DNA. Base pairs. T is going to go with A again. And G pairs with C. A pairs with T. It's pretty repetitive, but it is good practice. And you should be, finding a rhythm by now. C and G, they kind of look alike, so they go together. A and C have more angles, so they kind of pair together. Whatever works for you to remember. C goes to G, T goes to A. This right here is my DNA, base pairs. So that is it. We have just created a full double strand of DNA, and this might be really important information for your hair color or something like that. And we'll get to a little bit more of that later. But hopefully you feel comfortable with that because you might be doing a little more practice with it on your own. Remember, C goes to G, T goes to A. This right here is my DNA. Alright, so now what? Like I said, we're going to learn a little bit more about DNA. It can't just stay inside the nucleus. So think about it. How can all of your cells get information if it's just inside the nucleus of each of your cells? It needs to move and do things. So your nucleus has your DNA and it stays in there, but other things need to be made. And those things are proteins. So proteins must be made because they're going to leave the cells and deliver instructions to body parts like muscles and your brain. So DNA has to be turned into proteins, and we call this protein synthesis. So our goal is to take our DNA, use its information. So you might be wondering, how is a protein made from DNA? And I'm going to explain. There are 2 parts to protein synthesis. I'm going to keep it as simple as possible, but part 1 is called transcription, and that's where you create a molecule of what's called mRNA from the DNA in your nucleus. Okay, so I'm going to get more into that in a minute. Just know that is the first part of making a protein from DNA. So this picture's a little crazy, but it does go over some really key steps to this process of what's called transcription. So the first thing that has to happen is your DNA, which is wound together like this, it has to get unzipped. So a protein will come and unzip it, meaning like kind of split the 2 parts apart. Then an mRNA strand, which you see right here, comes in and sort of copies that DNA that's now unzipped. So this mRNA is going to match with the DNA here, and then something cool is going to happen. So the DNA unzips, the mRNA is created that kind of matches with the DNA. So mRNA is just a single strand. It's not two like DNA. And all the bases are the same except for one. So there's no T in RNA. The T is actually replaced with what's called uracil, or a U. So instead of T going with A, it's going to be A's going with U. So I know it's a little confusing, but I'm going to show you with this practice right here. So you have your DNA strand, that's this one, and then you have an RNA strand that's going to pair up with the DNA. Okay, so this DNA has just been unzipped. The RNA is going to get created, and you'll see why in a minute. So now we have to create the right RNA strand that matches with this DNA. So C still goes with G, nothing has changed there. T is still going to go with A. There still is A in RNA, nothing has changed there. So you can still have A's being created. C will still go with G. Now this one is the special one because you have A. Normally you would say A goes with T, right? Because that's what the song says. That's wrong, okay? So there's no more T. A now goes with U. C still goes with G. Pretty simple. And a lot of kids get tripped up. T won't go with U. T will still go with A. Okay? It's just A. Once A is in DNA, it makes a U. That's the only confusing part about RNA. Everything else is the same except A goes with U. I don't have a song for that one. You just got to remember it. So, like I said, protein synthesis has two parts. We just learned about part one, which is transcription, which is creating a molecule of RNA from the DNA. Okay, so that's one part, part one. So you have your DNA, and then it's transcribed into RNA right here. The next part is translation. Translation is really where you kind of change it all together. So you take the RNA that's been created, and you produce these specific amino acids, which we'll get more into in 1 minute. But essentially those amino acids make up proteins. So you have transcription, then you have translation, which makes the RNA into proteins. Okay, so you take a specific mRNA like the one we just worked on, and that will become a protein. Okay, so this strand that we just had here would then code for specific amino acids that become proteins. So we're going to go back to this picture again just to review some of the differences between transcription and translation. So translation, you have the RNA, and it's actually going to leave the nucleus. So this doesn't happen in the nucleus. The mRNA leaves and goes into the ribosome. If you remember from way back when, ribosomes make proteins. So remember, it's kind of way back when, but ribosomes make proteins. So the RNA has to go into the ribosome for that protein to be made. Okay, and that's what you're seeing here. And I'm going to break it down on the next slide. Okay, so the steps are a little confusing, but we've gone over most of them. RNA moves from the nucleus into the cytoplasm and then attaches into the ribosome, which is this gray thing here. So the RNA hooks itself into the ribosome. Then something called tRNA comes in and kind of makes a little code for specific parts of RNA. Okay, so this little section of RNA that's been created is going to code for a specific piece of tRNA. Okay, so your tRNA are these big molecules here. They come in and they read the mRNA. Sorry, my drawing is so bad. And then they bring an amino acid, which are these guys up here. They bring this amino acid that will match with this specific piece of mRNA. So this amino acid will then be the perfect match for this section of mRNA. And then it does it again for this section of mRNA until you have a chain of amino acids like this that will then form a protein. And that protein will then leave the ribosome and go do work in other parts of the cell where it's needed. So remember that it never just stays in the nucleus or the ribosome. The protein will go where it's needed the most in the cell. So that's the whole goal of protein synthesis, is to make these proteins that can then help other parts of the cell using the DNA. So that is translation. And just in case you're wondering, amino acids are these building blocks, so they're what proteins are made of. So this big chain of amino acids will eventually form into this big protein here. So proteins are made from amino acids, which are much, much smaller, and they build into form proteins. And again, those amino acids are formed by using mRNA strands to then create an amino acid that matches with it. So just to review kind of everything that we've gone over, here's my DNA song again. But remember that T still goes with A when we're talking about RNA, but A now goes with U. And then the major steps that we've learned today are transcription, which is where you take DNA and create RNA, and then translation, where you take RNA and create proteins. So those are the major things you should have learned today. If you have any questions, please let me know. Have a wonderful day. Stay safe.